Good evening, everyone. I, Tejaswi Kamath from the Public Relations Office, welcome you all to the celebrations of National Science Day at IIT Bombay. National Science Day is observed every year in honor of Sir Chandrasekhara Venkat Raman, an Indian physicist who discovered the Raman effect, for which he was, as a part of celebration, the institute has organized two lectures. The first session will be taken up by Professor Samir Maji from the Department of Biosciences and Bioengineering, IIT Bombay, on mechanism and blood test development of Parkinson's disease. The second session will be delivered by Professor Varun Bhalerao from the Department of Physics, IIT Bombay, on ultimate alchemy, how does the universe make gold? Beginning with the introduction of our first speaker, Professor Samir Maji is a faculty member in the Department of Biosciences and Bioengineering since 2009. He was appointed as an institute chair professor in 2022 and is also the first professor in charge of Sunita Sanghi Center for Aging and Neurodegenerative Diseases at IIT Bombay. He pursued his BSc and MSc in chemistry from University of Calcutta, West Bengal. Thereafter, he pursued his PhD in peptide chemistry from Indian Association for the Cultivation of Science, Jadavpur, Kolkata, West Bengal. He has done his postdoctoral studies from Harvard Medical School, Boston, USA, University of California, Los Angeles, Salk Institute, San Diego, and ETH Zurich, Switzerland. His research interest primarily includes studying amyloids associated with human disease and native functions. Professor Maji is a recipient of numerous awards and distinctions, including the J.C. Bose Fellowship awarded by SERB India 2023, editorial board member of BBA Proteins and Proteomics Journal 2023, the Sastra Obed Siddiqui Award for Life Science 2023, the Professor S.C. Bhattacharya Award for Excellence in Research in Pure Sciences 2022 for his outstanding contributions in the area of protein aggregation, biochemistry, and biophysics. He is also the Fellow of Indian Academy of Sciences, Indian National Science Academy 2023, and National Academy of Sciences, India, among many more. I now invite Professor Maji to deliver his lecture.
This is very important day for all of us, that when we can imagine that early 1990s, when nothing was there, no science infrastructure, that C.V. Raman, who started his science at Indian Association for Cultivation of Science, actually where I did my PhD. And of course, this is the day, 28 February 1928, he discovered the Raman effect, and for that he got a Nobel Prize 1930s, and 1987 onwards we celebrate 28 February as a National Science Day. So I'm going to talk about that human bodies actually is very complex systems. If you think about that, almost we have 100 trillion cells and 200 different type of cells in our bodies, and 80 known organs and 20,000 different genes. So 20,000 different proteins in our body, and if you can think, a cells hold almost 42 million proteins molecules. So why I'm telling all this number, it tell you that how complex our system is. So if you imagine any circuit, somewhere one point gets defected, entire circuit don't work. So now you think about that 100 trillion cells, even you cannot afford one cell to be defective. And a single cell cannot afford even one protein cannot be defective. That means either you have to repair or you have to degrade. So cellular system cannot bear with any defective genes, any defective cells, any defective proteins. And I will tell you that how difficult it is if you could think. Let's say this is the kind of cartoon picture of the cells. So many different organelles, and all organelles actually they coordinated each other, do the functions. Probably that's why you are surviving. So now inside the cytoplasm somewhere you have a ribosomes which synthesizing the proteins. And if you think about this is the cytoplasm, it's like a gel, this huge crowding. So that means when protein gets synthesized, and this is the kind of polymers made by 20 different amino acids, you can imagine this will be that huge difficult job that protein have to get folded. And unless protein get folded, it cannot do the functions. So that means that interhuman bodies consist of all the cells, and all the proteins have to be properly folded to do the functions. So now this is kind of a rough pathway how protein gets folded. So that means whatever protein every single cell synthesize, huge number, actually they have to be folded by different pathway. That means there are distinct steps are involved. And within this distinct step, there is always possibility that protein get misfolded and make these kind of robust, lowest energy minimum structure is called amyloid. I will tell you what are these. So amyloids are protein peptide aggregates, 2 to 15 nanometers in diameters and up to several micrometers in length. Highly ordered, stable beta CD structures. That means this beautiful structure, if you can imagine, this is the most stable structural form of any proteins. And almost every protein have a capability to go to this form because this is the lowest energy minimum structure. So with time, almost 3 dozens of human diseases discovered associated with protein aggregations: Alzheimer's, Parkinson's, mad cow, type 2 diabetes, Creutzfeldt-Jakob disease are among them. So I'm going to talk about Parkinson's disease. These are chronic progressive neurological disorder, and actually the patient suffering with Parkinson's disease, they will have tremor and very slow movement, and a lot of time in extreme cases they will have completely loss of physical movement, is called akinesia. And if one look at the pathology, there will be Lewy body formation in dopaminergic neurons, and in dopaminergic neurons, actually whatever the surviving neurons, they become sick, they cannot release the dopamine, and you know the dopamine is essential for our movement. So Parkinson's disease, if someone look at the history, actually Indian used to know 3000 years back what is the disease, and amazingly what is the drugs also. So if you look at in Ayurveda text called Susruta Samhita, written by Susruta, it terms as a Kampavata in Sanskrit, that means Kampan, and people used to get treated with natural products from mucuna prunes. So this is the mucuna prunes, this is the seeds. A lot of people say is kind of black coffee, and these are full of L-Dopa, and today L-Dopa is the drugs. So that means we used to know 3000 years back what is the disease and even what is the drugs even. And if you look at the entire history, now we rediscovered I would say, that medical researcher called Galen, he described the symptoms, tremors while at rest. And then James Parkinson wrote an essay on the second palsy. So that's why the later actually people discovered that it is associated with basal ganglia. And then if you look at that, Jean-Martin Charcot actually gives called clinical lectures on disease in nervous systems, wrote a book, and actually he proposed the name called Parkinson's disease. Then during time that Frederick Lew discovered the Lewy bodies as a pathology, and you can imagine now people knows what are the genes involved for the Parkinson's disease. Alpha-synuclein SNCA genes, one of the important genes involved for the Parkinson's disease. And people actually when they look at Lewy bodies, they found that alpha-synuclein protein is the major component in the Lewy bodies. And then people discovered that alpha-synuclein aggregations and oligomerization actually essential for the cell death that happened in the Parkinson's disease. Of course, there is a lot of alternate theory. Some theory include that influenza infections was one of the major cause for Parkinson's disease. Of course, didn't get a lot of attention, but after COVID, a lot of people believe that Parkinson's disease prevalence will increase significantly due to the long COVID. So a lot of people believe that actually viral infections have a direct role for Parkinson's disease. So this is the Lewy bodies, and when someone isolated from postmortem brain, they look at that what about the amyloid fibers. You can see this is the alpha-synuclein amyloid fibers, and that's the beginning of the basic science, how to study the Parkinson's disease. And during time, people are able to kind of clones, synthesize the protein in E. coli, and when they studies, they found that amyloid, the structure, and that structure actually closely resembles to the Lewy bodies in the patient. Of course, during time, people study the mouse, Drosophila, different animal model, including cell cultures. And all these studies actually support the idea alpha-synuclein polymerization is essential for Parkinson's disease. And this is one of the cartoon pictures is showing that alpha-synuclein aggregations is important because metal, pesticides, all the drugs molecules which have a direct influence for Parkinson's disease can influence the protein aggregation of alpha-synuclein as well. And almost there are 10 mutations discovered so far for Parkinson's alpha-synuclein. And all these mutations either slow down or promote the aggregation of alpha-synuclein. So that means most of them, they are early onset Parkinson's. And I was part of the team. We proposed for the first times the not amyloid fibers for alpha-synucleins, but oligomers formed by alpha-synucleins actually direct relevance to the Parkinson's disease. And it's got huge attention, and after that, a lot of people look at that probably oligomers are the most toxic species, not the amyloid fibers. So another problem with the Parkinson's disease is infectious. That means that in the some part of the brain when it starts, with time, actually spread all over the brain. And not only that, a lot of people believe that it can transmit literally from one human to another human. There is a possibility. So when people did, for example, brain homogenate inject to the mouse, that mouse become Parkinsonian mouse. So therefore, there is these all amyloid disease actually people propose they could be infectious as well. So Parkinson's disease in India, I will say that we are not progressing well due to many reasons, because we have a lot of prevalence of infectious diseases, and Parkinson's disease is late onset, and of course that a lot of people used to believe that we take Indian curry spice, therefore we have a less prevalence. But if you can imagine that Indian population used to have life expectancy around 38 years during independence time, now is close to 72. So that means 1% population above age 60 can have a Parkinson's. So we have a 7 million now. And if you think about 30 years later, our 70% population will be kind of, aging population. So we'll have a huge burden for the Parkinson's disease. And that's why you can see that India is one of the most red zone according to WHO, that we are going to have Alzheimer's and Parkinson all will be affected. India will be severely affected. So this is the synuclein sequences, human sequences. We have 3 different proteins. Alpha-synuclein is wild type, beta-synuclein, gamma-synucleins, and their role not clear, but people believe that alpha-synuclein have a role for vesicular transport. But if there is a mutation, for example, as I told you, there are almost, so here I written down 7, another 3 discovered recently. All familial mutation of alpha-synuclein actually altered the kinetics of protein aggregations. But very interestingly, if you look at age onset of the disease and lag time of aggregations, there is no correlation. So that means you are going to expect that if there is a mutation, they should promote aggregation if aggregated species is the toxic species. But that is not the case. Some of the mutant, they actually slow down the aggregations. And that's why the probably oligomer things comes into the play. We hypothesize that sustained population of oligomers, whether they are faster or slower, this oligomer actually making much more prone for toxicity by the mutant proteins. So it is not clear here. So we did a lot of cell biology experiment, and using dot blot against A11 oligomer specific antibody, we were able to show that actually mutant in presence of dopamine and rotenone, these are all compound promote Parkinson disease. Actually, they are making much more oligomers compared to wild type proteins. So that means that mutant protein is much more susceptible to the chemicals which promote Parkinson disease to make the oligomers. So this is kind of hypothesis we propose, that mutant might promote oligomers formations or make sustained population of oligomers. That's why they are making early onset Parkinson's. So then we did the Drosophila experiment in our lab. So here in the Drosophila is the fruit flies. So here we introduce the wild type and mutant alpha-synuclein. You can see that E46K, this is the faster mutant, H50Q faster mutant, G51D the slower mutant. But if you look at the data, their survival, you can see that both wild type and this is the control, all three mutant actually flies going to die early. So then we did a lot of biochemical experiment. We were able to show all these mutant, irrespective of faster and slower, they're actually making much more oligomers. And these oligomers actually responsible for the toxicity in the Drosophila. So now also we asked during time that alpha-synuclein concentration in cells is lots, but it's not millimolar or huge amount. But somehow if there is a local concentration increase, what happened with the alpha-synuclein aggregation? We were able to discover that alpha-synuclein actually form hydrogels. And these hydrogels, if cell exposed to these hydrogels, actually there is a massive cell death happens. So then during time we were able to show this hydrogel actually work like a depot for slow releasing toxic oligomers. You can see here the dot plot with time. You can see that with time that these alpha-synuclein hydrogels actually releasing the oligomers, and these oligomers are highly toxic. So basically we propose that if somehow local concentration become very high inside the cells, alpha-synuclein form hydrogels and that work like a depot of oligomers, and these oligomers actually kill the cells. And now there is another field evolved in last decades called liquid-liquid phase separation. I will tell you why I'm introducing here in Parkinson's disease. So as you know that if you put the oil in water, right, most often we see that oil phase separate from water, right? Because oil cannot mix with water. And this is the video that how they phase separate. So during time, people figure it out that there are many organelles inside the cells. They don't have a boundary. That means they don't have a membrane. So that means people are wondering, if there is no membrane, how they can do the functions? Because cellular nucleus, they covered by membranes. That's why nuclear reaction should happen inside the nucleus, not in the cytoplasm. So membraneless organelles, how they form and how they function, people figure it out. This is actually process of phase separation. And when the biomolecule phase separate, maybe proteins and nucleic acid, they can make is called membraneless organelles, and these organelles actually do the functions whenever need, and then whenever cell don't need, they can dissolve them. For example, if cell gets stress, they make a stress granule. It's work like organelles. When stress removed, stress granule dissolve. So that is the good part. It's like you are making tent in forest, and you cook, you eat, and whenever your job is done there, you can come back to home. There will be no tent. So recently in 2020, we discovered that alpha-synuclein make a liquid-liquid phase separations. This is the fusions you can see. And if you do the PrEP, they recover very well. And they also getting little bit rigidified even after phase separations. Very interestingly, if you can see that all the conditions which promote alpha-synuclein aggregation, these are all Parkinson-associated conditions, metal exposures, familial mutations. We are able to see actually they promote the phase separations. And this is the PrEP assay, this is the recovery assay. You can see actually they rigidified right away. Lot of kind of conditions, they rigidified the liquid droplet right away. And this is the video. You can see they form, they dissolve, and a lot of times small droplet they fuse and make a larger droplet. And these larger droplet, they rigidify and actually bursting the amyloid fibers. And that's why we propose the hypothesis that these droplet rigidification actually make the amyloid fibers, which eventually make hydrogels. And we did a lot of cell biology work, and we were able to show that inside the cells, alpha-synuclein also form phase separated state, and with solidifications, actually they make amyloid-like appearance. And later, other group from UK, they also showed that in C. elegans, alpha-synuclein do phase separate and then make liquid to solid transitions. So this is our kind of proposed mechanism, and this makes that huge difference in the field, because a lot of people now doing work in these directions, that alpha-synuclein amyloid aggregation might happen through the liquid-liquid phase separation. So now I will discuss about another dimension in our lab we work, that the Parkinson disease, the clinical diagnosis and treatment. So if you look at that male, actually that male have kind of more prevalent in Parkinson disease compared to women. And somehow, for some reasons, Indian have even much early Parkinson compared to Western world. People don't know. So of course, the etiology of Parkinson disease is not clear, and there is no biochemical test. Basically, there is no test, clear test can tell Parkinson disease. And very important thing is that when somebody appears with the Parkinson's, is almost stage 3. So that means stage 1 and early stage 2, it will be very difficult to figure it out whether somebody has Parkinson's. So that means when somebody go to stage 3, that means that your course, whatever that drugs you are going to take, drug doses, it's getting very, very difficult. So very essential, the diagnosis of the Parkinson's for the single test, and early onset diagnosis is very important. Of course, there is no clear medicine which can cure Parkinson's, but Parkinson disease alterations or you can actually stop the symptoms, or lifestyle modifications can make a massive difference in Parkinson disease. For example, this is the PET scan. So fluorodopa PET scan, you can see clearly normal versus Parkinson disease. But the thing is that, so fluorodopa PET scan, normally people will not do unless there is a clear symptoms will appear. So nowadays, a lot of people are doing also dopamine transporter scan called DAT scan. But all these people do at the late stage, or it do not clearly distinguish between normal to Parkinson's all the times. So there is another test is evolving in the field. It's called protein misfolding cyclic amplifications done by Claudio Soto. So using this test, actually they come up with the prion disease, it's called SCAPI, detected from the blood. Of course, that during time, they are also applying that whether they can detect for Alzheimer's. So the entire PM says that based on the principle discovered by Stanley Prusiner, he got a Nobel Prize in 1997, where he proposed that proteins have, this is the prion proteins, have distinct structure with time or due to some mutation, that it's changed to these structures, and these structures work like a infectious particles. It's like a virus. It can replicate. It can go from one cell to another cells, one species to another species. So imagine that somebody taking meat, and this is, let's say, mad cow infected beef, and somebody take the meat. So that means that if it is infected, goes to the human, the prion particles will cross all the digestive tract, cross the blood-brain barriers, go to the brain, and now convert normal prion protein to the infectious or scrappy prion. So it's work like a virus. So based on this, we propose that the blood test of Parkinson disease development almost, I would say, more than a decade back. So but the problem with alpha-synuclein is unstructured proteins. So if you do the test, which is essentially required incubation sonication, that wild type alpha synuclein, which is unstructured protein, automatically will give the false positive. I will show you here. For example, this is the misfolded seed. Let's say Parkinson patient blood serum will have misfolded seed. Now, if you incubate with the normal proteins, wild type alpha synuclein protein, whether seed there or not there, you will get the false positive. This is the non-seeded aggregations, you will get a false positive data. So that means that wild type alpha synuclein cannot be used for the protein misfolding assay. So what we did, based on structural analysis, lot of structural biological study have been done. We designed two mutant, and that was very critical. And we were lucky as well that we got two mutant, A46P and A53P. And you can see that wild type and A30P, they are aggregating. These two mutant don't aggregate. And if you look at their electron microscope and AFMs, they don't aggregate as well. So we are very happy that at least these two mutant not aggregating. And we know the reasons why they are not aggregating, because their protein-protein interactions, if you look at wild type and wild type E46P, they are working. But if you look at E46P, E46P, A53P, they are not interacting. So basically that this mutant, they could not able to interact each other. That's why they are not aggregating. But once you give a seed, look at here, that this is your wild type and this is your E46P, A53P. If you add 1% seed, that means infectious particle that's supposed to circulate in the blood. If you add, now proteins is aggregated. So this is kind of eureka moment for us, that this is working, system is working, now we can establish the assay. So based on this, what we did the simulated serum. Basically we took the healthy control serum and we spiked with the lab generated seed, and this is the actually the principles. So we add, this is the proteins express and purified from E. coli, and then we did incubation sonication cycle, and then you would see the single red, actually they are multiplying, and then eventually if you centrifuge, you will isolate this and you will analyze this. So now a single seed which cannot be analyzed here, now you have millions of these, so you can analyze the data now. So based on that real experiment, when we did, you look at here alpha synuclein fibers, and what we got, that even femtomolar nanomolar scale, we can actually amplify, and you can see the data here after 4th round. Not only amyloid fibers, even oligomers is working. Did you see that oligomers, they are slow, but fibers is very fast. So basically we were able to show that with this mutant, which do not aggregate alone, but if Parkinson patient blood serums have femtomolar concentration, oligomers or fibers, we were able to detect. So this is the principle. So then we did the concentration dependent analysis, that what is the lowest concentration you can look at 10 to the power minus 15 molar concentrations, we can able to detect that. So then we started the clinical trial. So basically that this is the inclusion criteria, the bradykinesia, muscle rigidity and postural instability, and this is kind of based on this we kind of recruit the patient. Actually KEM did based on the UK Parkinson Disease Society, and this is the all kind of data, that what is the disease durations and what is their disease scale. So then initially we did trial with the 12 different patients, and some of the data you can see clearly that, let's say this is PD1, PD2, PD3, you can clearly see that after 4th round, if you look at that PD patient, Parkinson patient, they're clearly very high compared to the normal human control. So that's suggesting that our assay is nicely working. But one problem I should tell you, that this Western blot analysis is very tedious, and it itself might take 2-3 days and a lot of manpower. So parallelly we established another technology, like can we put that called blot and then do with the congruent. Congruent known to bind amyloid, and then you can see that what happening that after was. So this is the before was for all Parkinson, and you can see this is the after was. So did you see that all the control, almost they don't see, you don't see the signal. But you see that afterwards, most of the Parkinson patients actually they are getting the symptoms. So now we completed 40 patient clinical test, and you can see that we are almost close to 93% sensitivity and close to 90% specificity. We will to kind of analyze the Parkinson disease, and now we are trying for 100 patient clinical trial. Hopefully that in future and within years or two, we are able to launch the test, and this will be the first blood test, I believe, in the entire world. And India, clearly we don't have, and we are very proud of this test. And if you look at that, the control when we did with tau and A-beta, with the Alzheimer's disease and IAPP for type 2 diabetes, we don't see this kind of amplification. So we believe these are specific to Parkinson disease. And then in future, what we are going to do is like a PCR, based on the number of cycles and how much amplification happens, we are able to back calculate and tell what is the initial seed concentration circulating in the blood. So then you can imagine that if there is a late onset PD, that means lot of seed will be circulating. So our amplification will be very fast. If somebody have early onset PD, the seed concentration will be very low. It might take long time or many cycles to amplify. So based on that amplification cycles and amount of the amplification product, we believe that we are able to tell that what stage of Parkinson disease and in future, early stage of Parkinson disease is possible for the detections. With that, I will acknowledge, of course, IIT Bombay, give all the support and scan center, and lot of also work going on with this center, and DST, Department of Biotechnology. They are very kind to give all the support throughout my career. WRCB, BIRAC, CSR, and Department of Science and Technology, and ICMR, and Tata Center for Technology and Design, IIT Bombay, where we got the seed funding for this blood test. I think almost, I don't know, 8-10 years back. And this is my lab, not very recent, but I would say that these are the pillars, and we have a big lab, and people work very hard, and that's why we are here. And specifically, I presented the blood test work done by Luxvikant, some support from the Urumina, in collaboration with the Sangita Rao from KEM. And now we are starting a huge amount of the clinical trial with different hospitals like Hiranandani and other hospitals in Mumbai. And also we are starting collaborating with AIIMS. So we believe that our blood test can make a difference in the society. Thank you very much.
Thank you, Professor Maji, for such an insightful lecture. We will now take questions from the audience.
Good evening, sir. Thank you for the lecture, and I'm very great that you have made one good story to the blood test. I'm not from this medical background, but I have a general question like at what age the Parkinson starts, and since early detection is not possible, so what are the maybe the level 2 symptoms or the earlier symptoms for the Parkinson's, and how we can avoid? Is it like that coffee we have to drink like yours? Okay, at the start. Very general question.
It's a good question. So basically that so more than 60 people, 1% population get affected for Parkinson's. So that means if somebody kind of 75 or 80, there is a 75-80% probability that person might get Parkinson's. And in India, we get a little bit early onset. So our average is around 55 or so. So 10% Parkinson patients are from kind of familial mutation, basically that they carry this mutation. They are genetics. 90% of Parkinson's, they're idiopathic, or that means unknown cause. So that means lifestyle and food habit, exposure of pesticides. So different things can cause the Parkinson disease. So early symptoms is mostly non-motor symptoms. So a lot of people now started working. So there, for example, sleep cycle, there is a change. Mood will change. So people get frequently angry sometimes. And lot of people think that lot of behavioral kind of non-motors actually functions at the early stage. So that's why people are trying to recruit the patient based on early stage symptoms. The thing is that it is not clearly established that a specific, for example, phenotype or behavior which can be linked, because there is called Parkinson plus, called Parkinsonism, is a group of disorders. So lot of symptoms they are overlapping. So it will be very difficult to conclude whether it is Parkinson or Parkinson plus or Parkinsonism. So therefore, that's why that early diagnosis is very important. For example, in future, one can imagine that age of 60, somebody every year go and do the blood test. If some clinical test come, so that they can take the mandatory one of the test, probably early diagnosis is possible. So now one can ask that we don't have a medicine. What is the point to have a diagnosis? So we have a very good modified drugs. For example, we have LARC inhibitors, we have a MAO-B inhibitors. If somebody detected early onset, so you should not take the levodopa. People generally don't take levodopa rather than take the MAO inhibitor. So basically that what kind of drugs, doses of drugs can make a difference another 3-4 years of life. So therefore that knowing that somebody infected with the or affected with the early onset Parkinson's is very important for the rest of the life, and it makes a difference.
And also even economical question, how costly will that test be for a general public or for any hospitals if one would like to go for such a very specific test?
Yeah, so these tests, frankly speaking, we don't know now. Right now we are calculating. Of course, we are also trying that can we make it high throughput. So if it is high throughput, it will be of course less expensive. Right now, the major problem is that is required the trained manpower and a couple of machines, instrument. We are also trying to make our own instrument. So therefore, that for all the corona, because is required the proteins, and protein is very expensive. So right now we optimize high throughput way, for example, a bioreactor, or so can we make the huge amount of the proteins with less amount of fund required. So we are trying to bring down as easy as possible. But right now I am not sure what will be the cost. We have not calculated it, but we are going to do soon, because probably people are going to ask what will be the cost. But I will say that right now if somebody want to do clear-cut results, that is Parkinson or not, it's massive expensive. You need 2-3 imaging techniques, lot of behavioral assay. And one of the pathetic part is that how Parkinson's disease confirmed when somebody take levodopa. It's somebody responds to levodopa. That means diagnosis is confirmed. So basically that somebody is taking medicine, if symptoms improve, you are diagnosing, oh, must be that disease. So all other imaging technique actually excluding the possibility of other diseases. So our major goal is, can we have a single test, can predict 90% probability that person is the Parkinson or Parkinsonism. Then we'll look at large scale clinical trial, and maybe we can find the early onset, because early onset recruit of the patient is difficult. So but the way we are going that in future we will do it. Yeah. Thank you. Yeah. Yeah.
Thank you, sir, for the valuable insights that you have shared with us. So these are the neurological disorders which were at the initial stage. But what has been observed during or post-COVID is people with mental health challenges. And to the best of my knowledge, we have very few professionals in this field compared to the patients who are undergoing this thing. So mental health challenges in terms of bipolar, schizophrenia, and many such things. See, the science is progressing very rapidly. We'll be living in a totally different world by 2030.
Right.
So do we have a permanent solution to mental health challenges, or how it will? Please guide.
So it's of course that is a huge challenge. Even I would say that for India it is a massive challenge, because if you look at that, we don't have. So if you look at that, so somebody affected with the Parkinson disease, actually there will be not many doctors are around. So that means that the kind of population we have, we don't have enough doctors who are very very much expert. And then you see that even in US, if you think about that, the complicacy, because this is a multifactorial disease, the complicacy that what kind of disease is. So you have to invest a lot of time and a lot of kind of money patient has to invest to figure out what the disease is. So I according to me that we are progressing very well, and so we have to look at the policy. That's probably our center is working, Scan Center. We are kind of targeting the policy, assistive device, and other kind of different angles, so that probably we can have some solution for the future for Inter India. That, for example, that this mental health issue is not always associated with fund or associated with money. I think that we have to look at the policy matters as well. So I think IIT can have a huge contribution. That's why our Center of Policy Studies is working.
So further to what you said, are these mental health challenges hereditary? 100% hereditary? How is it?
No, so mental health is, for example, somebody have a dementia, somebody have a depression. So these are all mental health, right? So basically these are huge spectrum. So as I told you that Alzheimer dementia is almost 10% are hereditary. So 90% are sporadic for unknown reasons. So I will say that stress have huge impact. Lifestyle will have a huge impact. What kind of food you are taking will have a huge impact. What kind of society you are living in. So according to me that India might have a solution, because I think we have, so if imagine that Inter India you can make small grid 5 kilometers versus another 5 kilometers. So you will find one grid, they might not have mental health or Alzheimer's or Parkinson's due to the food habit, due to the lifestyle, due to the culture they have. So I will say that India will have a huge opportunity to figure it out, that what could be the cause, what could be the solution. But the problem is very expensive. It's not like somebody measuring the temperature. It's not malaria that any nurse can measure the malaria test. It's multifactorial. It's very difficult to access. Even Parkinson, whether Parkinsonism versus Parkinson is very difficult to figure out.
So my cousin she has graduated in bioscience, and I happened to discuss with her about this term called as CRISPR, CRISPR.
CRISPR.
Yeah. What I was of the opinion that this can alter the passage of the inheritance that is there from the ancestors, and this can stop. But what she told me is it is not legal in our country, and it cannot be done on humans.
No, right now I think it's not right now legal, illegal. That is also time, right? Time. For example, in future there is a possibility. The thing is that for these kind of disease, as I told you, society and what kind of stress you have, or what kind of food you have, what is lifestyle you have, that will probably dictate. So this dementia or kind of mental health, right? So therefore, genetics will be very little involved. So even if you do CRISPR-Cas, remove whatever the gene, 90% no guarantee that person will not get. So that means that 90% probability that person will get. So then I don't know what CRISPR-Cas will do. So I will say that it's mostly 90% are kind of sporadic disease or idiopathic cases. So that means that it will be very difficult to find solution very soon. It's a long way to go. Yeah. So maybe later, so let's start. What do you?
Yeah. So my question is that you mentioned about a mutation A35E that kind of delays the toxicity. So my question is that while you were kind of doing these assays, did you find anything relating to this particular mutation that?
No, that is an artificial mutant. That is nothing to do with toxicity, nothing to do with anything. We want to develop this blood test. That's why we use. We need a mutant that will not aggregate. You do sonication incubation for the entire process. we need a mutant that will not aggregate unless there is a seed from the patient. And that actually set up the basis for the blood test. So this is artificial mutant generated by us, designed mutant.
So can this be kind of thought that if we, as you told, that we still don't know how we are going to treat this disease, and this kind of this artificial mutant be thought of as a kind of future prognosis for the disease?
No, but as I told you that only 10% cases are genetics, right? 90% are sporadic, I told you. So that means that, for example, if person A, there is no genetics involved, no mutation, still there is a 90% probability that person might get Parkinson's. So it's only 10% are linked with genetics.
So just a sincere request, means need a feedback from you of people with mental health challenges, like from what you have explained to the modern day, which challenges the people are facing. So how do organizations consider these cases, and how about the environment of people with mental health challenges?
I think awareness is very important, and we need some kind of social structure. I can tell you, somebody was telling in US, for example, or that is true for here also. For example, somebody retired. Let's say after retirement, even they need to be more engaged. Otherwise, they will have a problems, right? Their engagement getting difficult. That's why I think our policy center, they are working that how they can be more engaged, so that they don't have even time to think other things. They will not get depressed. So I will say that it's from all angles. For example, let's say somebody have a Parkinson's or Alzheimer's. So Alzheimer's person, according to me, should not stay at home, because sons and daughter cannot properly take care. They should go Alzheimer home. They can take care very nicely. But where the home? We don't have a home. I will say that we are progressing very well, and we need now more multi-angle effort from different, in terms of society, organizations, all the people, because everybody will age, and think about that how to tackle these problems. And India will have a problems, and we have to think that problem is coming. Recognition is most important. So that's why I showed the data. Alzheimer's, Parkinson is less recognized disease in India, because we are kind of dealing malaria, all kind of infectious diseases here. But it will be very dangerous time. Another 10 years later, we will have a huge number of Parkinson, Alzheimer's. And almost right now, every day I heard his family, that family, they have Alzheimer's or Parkinson's. So I think somehow we have to think in terms of policy, infrastructure, blood test, everything we have to think that how to sample.
Requesting your guidance on lithium drugs.
Actually, I cannot guide you, but what we'll do that we do organize some talks in our center. So maybe you can keep in track our scan center if you can do.
Hello.
Yeah.
Sir, as alpha synuclein is present in both like Parkinson's disease and as well as in the Alzheimer disease. So will this test can distinguish Alzheimer's from Parkinson's?
Yeah, because in Parkinson's disease only alpha synuclein get aggregates, right?
Yes.
Alzheimer's another protein get aggregates, right?
Yeah.
So therefore, the aggregates circulating in the blood, alpha synuclein aggregates circulating in the blood will happen only for Parkinson's disease, not for Alzheimer's, I believe.
Okay.
But we did the experiment in vitro, you can see, but we are now going to do the patient trial. Basically that we have to take the blood serum for Alzheimer's patient and take a look whether this assay giving false positive or not. So we can distinguish Parkinson's versus Alzheimer's. So that study need to be done before test got launched. So we are going to do now.
Any more questions? If we have no other questions, we'll move on to our next session. Thank you, Professor Maji, for the lecture. Moving on to our next session, I'll begin with the introduction of a second speaker, Professor Varun Bhalerao. Professor Bhalerao, an alumnus of IIT Bombay, acquired his B.Tech degree in electrical engineering in 2006. He then pursued his PhD in astrophysics at Caltech in 2012. As an astrophysicist, he primarily studies transient astrophysical sources like supernova explosion, gamma ray bursts, etc. At IIT Bombay, his group works on the development of Growth India, that is, India's first fully robotic telescope. The group leads the Indian effort in the study of elusive electromagnetic counterparts into gravitational wave sources. He leads the ambitious Daksha project to build the world's most sensitive transient space telescope. Professor Bhale Rao is a recipient of various awards, including Vainu Bappu Gold Medal, the highest honor given by the Astronomical Society of India, IIT Bombay Early Researcher Achiever Award 2019, the Kriti Ramamritam Award for Creative Engineering 2019, IIT Bombay Departmental Award for Excellence in Teaching 2023. He is also an associate of the Indian Academy of Science and is a recipient of the DST Inspire Faculty Fellowship. I now invite Professor Bhale Rao to deliver his lecture.
All right, so I'm Varun and we are here for Science Day. So before I get into my topics, let's talk about Science Day. Okay, so Science Day itself is meant to remind us about science and doing science in India, and the focus of this year is actually science for development. Okay, but we have to remember that science is something which is not an activity we do in a classroom or in a lab for 5 minutes, 10 minutes, whatever, and then forget about it. Or occasionally forget about it while we are doing it in the lab, right? That's not what it is about. It's actually a constitutional duty for us to have what is called a scientific temper, okay? And what it means is that you actually have to think about things scientifically. You have to go for evidence-based knowledge, evidence-based understanding. And actually in that very spirit, don't take my word for this, right? You go, and the Constitution actually is a surprisingly small book, okay? Go look this up, go read it, right? And of course, if you are feeling bored about reading a book that you suspect might be dense, you can also start off simply with Wikipedia, like we always do. And it will also tell you about scientific temper, but there's a pretty cool opening line that Wikipedia has. It says scientific temper is a way of life, right? So science is a way of life. And today on Science Day, as we are hearing talks and we are thinking about C.V. Raman and so on, let's remember that science is broad, science should be all encompassing, and it is our fundamental duty as citizens of this country. With that, let's get started. So here's a question for everyone. Apologies to online listeners because I can't get their answers, but what's your favorite element? Iron, okay. There's someone with iron. I'll give you 1 gram of iron after the talk. What else? Gold, okay. Immediately jump to gold. So can I take away all the oxygen in this room? Already silent even before the oxygen is gone. But yes, humans are funny, right? We just heard a talk about proteins and everything. We are talking about carbon, nitrogen, oxygen. First answer was iron, second was gold. No organic chemistry here, okay? Fine, but yes, indeed it is usually this that we lust after, right? We want gold, we want piles and piles and piles of gold. And in fact, the contrast is still not great here, but that's okay, I guess we'll make do. In fact, alchemy is essentially this field that has been existing for a long time, when people did funny things to try to make gold. Typically gold from lead. I have no idea why they really hated lead, but they didn't want to convert iron into gold or anything. They always wanted to kill lead and convert it into gold. But by now we know a bit better. We know we can't actually go around and make gold like this, but yet gold exists around us. I mean, the fact that we know that gold is valuable means that gold exists. Otherwise, you wouldn't have value for it. So how do we do this? How do we get all this gold around us? And to do something like this, of course, you have to start from the very beginning. So you have to go and start with the Big Bang, okay? So we can get started here. Not this kind of Big Bang, not this Big Bang theory, but the Big Bang theory which created the universe, not the theory, the bang itself. And it began out of nothing and essentially created a universe that was largely hydrogen a little bit of helium and some trace amounts of lithium. So if you are a chemistry student 13 billion years ago, life was very easy because you needed to know hydrogen, helium and lithium, that's it. Right? This is also where IIT and counting lingo comes from. There is 1, 2 and infinity. Right? There is hydrogen, helium and everything else. Astronomers also use hydrogen, helium and metals. Okay, so for astronomers, carbon, nitrogen, oxygen are all metals. But this is quite a lame periodic table. We can't give him his gold or give him his iron. So we need to start from here and we now need to forge the elements literally, not in a metaphorical sense that you would use in say Lord of the Rings. Right? We really have to get started from this. Build your own universe kit, khareed ke laye. It was delivered to you today. Now you have this universe which has only pretty much hydrogen. So what do? Well, the short answer is you simply wait for long enough, because hydrogen is a colorless, odorless gas which given enough time turns into people. Because this universe which started off with just hydrogen indeed did result in us sitting here and me talking and you listening to it. There are subtle details that we do have to worry about. So what you have to do first is you have to take all of that hydrogen and make stars. So here's a big blob that started off just as a blob of hydrogen, and then as it collapses under gravity, you can see that there are interesting structures that are forming here. Okay, things which are looking yellow are higher density regions, and as you can see, we will zoom in here and you will soon start seeing white dots appear in this picture. Okay, so there is one traveling out over there, here are a few down here. All of these are stars. Whether this doesn't happen quite this fast, turns out that the patience the universe has is a bit more than the patience we have. So here I am actually taking this very fast. Every second is 3,500 years. And you can see a large number of stars that are being created here. Okay, these are the first generations of stars that were formed, and as you form stars, they are interacting with each other gravitationally, they get thrown out of the cluster, all sorts of interesting things happen. This also tells you something interesting, by the way. This is a real simulation of star formation. Most stars in the universe are actually formed in groups or clusters. Okay, so the sun being a lone star sitting is not the most common scenario. More than half the stars are actually in groups or clusters. Okay, so this is rare. But now inside each of these stars is where the universal alchemy really is happening. Okay, so what stars do is they shine, and the way they shine is by producing energy from fusion. Okay, so you start with hydrogen, you fuse hydrogen into deuterium, into helium-3 and eventually into helium, and in all of these processes you are generating energy. We hear about this in terms of fusion in stars, we also hear of this in terms of fusion bombs, but essentially, I mean, okay, gold kidhar hai, right? We started from hydrogen, we went to helium, Big Bang already gave us some helium, ye kya kachra hai? But no, kachra nahi hai, it has a lot of potential, because we can keep this cycle going on and on and on, okay? So we can take now this helium and we go through something called the carbon, nitrogen, oxygen cycle, and actually the only reason I kept this slide is all of organic chemistry comes in a single chain here, or all the building blocks. A lot of the organic chemistry follows, of course, is in great detail after this, but all those building blocks are being made in a single cycle that is happening inside stars. But that's going into too much detail, right? You didn't come here for a hardcore nuclear physics lecture, so let's simplify this. So what is happening is that at the core of a star, where the pressure is high and the temperature is high, you are essentially squishing atoms together so hard that they fuse into each other. And in the process, they release energy. Okay, once they release energy, what's going to happen is essentially you are now adding fuel into the fire. Or adding fire into the fuel in this particular case. All the fuel was already sitting there. Now when the pressure and temperature are high enough, you can take all the helium that was being formed in the center and fuse that to make carbon and nitrogen and oxygen, and then they just start sitting around there. Okay, now I have carbon, nitrogen, oxygen, then what? But then there is more weight, there is more pressure of the star coming in, so those now start merging and forming neon and magnesium. And from that you start going into silicon and sulfur and all the way up to iron. Okay, so you actually end up with stars that have this onion-like structure, right? At the top you see this harmless ball of hydrogen, and then as you go inside, as you start digging in, first you get burnt because don't go digging around in stars, but somehow if you manage to survive magically, then as you start going further and further in, depending on the weight of the star, you will cross many of these layers until finally you reach iron. Okay, and iron is essentially where the stars get some sort of a food coma. So what is happening is that you have energetically, when you squish these atoms together and when you create the next level, next element, you keep gaining energy until you reach iron. And at that point, the atomic nuclei are like, ab bahut ho gaya, I can't really eat anymore. So you are just full and you get saturated there. Okay? You can't create elements beyond that. And what that means is essentially what we say metaphorically when you go to a shadi lunch or dinner and you have your aunts or uncles saying और थोड़ा ले लो और थोड़ा ले लो, you say okay अब और नहीं हो पाएगा मैं फट जाऊंगा. Turns out stars take this quite literally. So you have a star that is happily shining around. The star will exist for tens of millions of years to maybe even hundreds of millions. These are heavy stars, they don't live for billions. But then in a matter of tens of milliseconds, okay, less than 0.01 seconds, this explosion happens. And this is called a supernova explosion. So the entire star just completely blows up. Of course, we don't see it in Discovery HD in real life. The way we look at it and the way we see it is something like this. So keep an eye on this galaxy, and what we are going to see in this galaxy is what a supernova was seen as by astronomers. So keep an eye on this circle. You see a star that is suddenly appearing out of nowhere, becoming brighter, and then stays bright, and over time you can see the date here, over time it is fading out. This is what we see a supernova explosion as. So here's a close-up. There was nothing there and suddenly something very bright came up. The explosion itself, as I said, happens in a fraction of a second, but you have a lot of material that you want to burn with that, right? So all of that peaking up takes time, and it actually takes about 2 to 3 weeks for it to reach peak brightness. So these are the supernovae, and with supernovae now you can start filling up the periodic table to levels where you can now start moderately hating memorizing it. Instead of having just your hydrogen, helium and lithium, you now have all of these elements, and you can see by the way there is a lot of color coding here. Okay, so exploding massive stars, these are the supernovae I talked about. They create most of the oxygen and arsenic and so on. A lot of these elements are created there. But there are other things as well. For example, a star like the sun is not spectacular enough to die as a supernova. The sun will die a wimpy death, and it will essentially, in that process, these dying low mass stars will create elements like this. Okay, a lot of carbon and nitrogen coming out of it. But, and technetium, by the way, is the last element here, which is all in a different color. This is so unstable that you actually can't create it naturally. Okay. So now you started off even with the supernovae and you reached only up to here. By at this point you might start wondering, okay, chal kya raha hai? How do we know all of this, right? I mean, astronomy in that sense is a very unique physical science. When we think of physical sciences, I talked right at the start of evidence-driven understanding. I can't, unlike other fields, go and say, okay, let me light up a star to see how it explodes as a supernova, right? So I want to observe objects, I want to study them, I want to analyze them, but I have no control over my experiments. So how do we do this? Well, we first said that, okay, we need to create a good network. So we started this network a few years ago called GROWTH, which stands for Global Relay of Observatories Watching Transients Happen. That's quite a mouthful. Astronomers like to make complicated acronyms. Okay, so in my group we often have competitions to come up with most weird acronyms. Some of them actually we stopped thinking of them as weird because we actually approve them later. So in GROWTH, what we did was we set up a network of telescopes all around the world, many existing telescopes, and then we set up a new telescope here called GROWTH India. Idea behind this network, by the way, is that you can imagine that the Earth is spinning around. You have day here, so you can't look at any stars. Suppose there is a star sitting up here. You can look at it from from observatories which are here. This one, although it is night, will not be able to see objects which are in this part of the sky. And then, so in our case, in this network, typically these discoveries begin with something called the Zwicky Transient Facility, or ZTF, in California. And as the stars start setting in California, they are now visible in Hawaii. So like a relay race, we hand it over to Hawaii. From there, we hand it off to Japan, Taiwan, India, Israel, Canary Islands, and then back to the US. Okay. And this Growth India telescope is this fantastic telescope located in Ladakh. Can we get these lights off still? Can we make it a bit darker? Is that possible? Okay, so this looks like a Mumbai sky. I was actually trying to show you how bad the Mumbai sky is by showing you this video, because you can see this band here, a very faint band. Still better than Mumbai, but not great enough. This is actually the Milky Way rising behind the telescope. Okay, so what you see down here is the dome of the Growth India telescope. Now, you'll see that there are lights flashing in that dome. You don't want to do that when you are observing. You are looking at sources which are very, very, very faint. But this video was taken when we were commissioning the telescope. So, when it was being set up, so they would take a set of test exposures, and with those exposures, then they would actually see if things were right, switch on the lights, tweak some things, and then go back to taking the next set of exposures. So they are trying to switch the lights off. Let's wait for a moment, and if it succeeds, I'll show you this video again. But meanwhile, here's what the telescope looks like inside the dome. This is a 70 centimeter telescope. It means the main lens is about this much. No stage lights. Maybe for a moment, even if I am in darkness, that's okay. So let's switch off the stage lights for just one moment, completely off. Much better. So let's look at this video again now. And that, by the way, is the difference of getting out of Mumbai and into good skies. Okay, so here's the Milky Way nicely rising behind the telescope. Quite amazing, isn't it? By the way, any of you who actually want to visit Ladakh, we do welcome visitors into this. Ladakh now has a nice Hanle dark sky reserve. So people are welcome. You can ping us and we will make sure that you can get a tour of the telescope itself. Of course, not at night time. At night time, they'll take you to smaller telescopes. Okay, thanks. We can have the lights again, please. Okay, so this is the telescope. This is the camera. It sounds very fancy when I call it a 4K by 4K high-end high QE CCD and so on. Those who are quick enough at multiplication will say that 16 megapixels, my phone has a better camera than that. Right? The difference is in these two words here, well, three words, but I've squished two of them into acronyms, astronomers. So high QE is all the difference. This camera is extremely good at capturing photons, extremely low noise, and it costs, phone costs have gone up through the roof, but this is still about 100 times more than the most expensive phones in the market right now. Okay, this camera itself costs something like 1.25 crores or some such number. Extremely high efficiency camera. Here's the dome, telescope sitting inside the dome, and you'll notice that the dome is a very uncomfortable place to sit. There's one plastic chair and a small table with random stuff lying on it. Looks pretty much like a hostel room, much worse actually. The dome also gets extremely cold, so nighttime temperatures currently are around -15 degrees. So you don't want to be sitting there, and the reason why it's uncomfortable is no one sits there. This is a fully robotic telescope. Here's, you have to have more pretty pictures in an AstroTalk, right? So here's the site. You can see the nice plateau. This is on top of Mount Saraswati in Ladakh. This was the first ever image that we took. Here are some more pretty pictures. I think Advait might be here, the student who did the coloring of this image from multiple images. But I said that this is not a telescope that someone sits there and operates. It's fully robotic. So here's what goes on behind the scene. Prepare for a 1-hour lecture on this now. Well, not really, right? But the point is that the back end is extremely complicated. This is the first and the only fully robotic telescope in the country, and all the automation was done by students of IIT Bombay, and that's a very important part, right? So it was all done here. This was work for a student's PhD thesis. About 10-15 undergrads have worked on it during this phase, and another 10-15 continue working on upgrading this, another PhD student, and so on. So lots of behind the scenes things. Here's a supernova as seen by our telescope. You can see that the object right at the center here is fading. Also, this is not as pretty a picture or a video as the previous ones, and this is by design. We actually adjust this to a very high contrast image because we want to see faint features, right? When you are studying something, you are interested in quantifying it, you are interested in finding things that are hard to find, you are not quite as interested in whether the picture looks pretty on your screen or not. Anyway, title of the talk was Ultimate Alchemy, so sona kahan? Right? So let's go and take a refresher here in terms of our gold making 101. So what we did was that we started with a universe, with a DIY universe kit. We got a lot of hydrogen. We formed huge hydrogen stars, and in those stars we started synthesizing metals. These are metals as named by astronomers, so anything above helium was a metal. Then those metals were still inside the star. So we had supernova explosions, we spread those metals around. Okay, these were still the first generations of stars. These metals now, when they spread around, those were not enough to achieve our purpose. So from the ashes of those stars, we had to form the next generation of stars. For example, we see that on Earth we have iron and gold and everything. Clearly, if they were being formed inside the sun right now, we would not have had them here. So how do we get those? So in order to get that, we need to, sun itself and the entire solar system is in that way reborn from the ashes of these first generation stars. So we make the next generation stars and we blow them up again. Now we talked about supernova. There is something called a supernova remnant. At the end of this explosion, what we end up with is either a black hole or a neutron star at the center. Okay, so we have taken some poor atoms, some poor elements, made them into stars twice, killed them twice. So for all you first person shooter fans, take that. Okay, and we are still not done, right? No gold. We were stuck to the first upper part of the periodic table. So how do we proceed from here? So can we make gold? Can we take a different approach? In modern times, instead of alchemy with just mixing random things and hoping to get gold, we can actually get things into a more interesting format with particle colliders. Right? So things like the Large Hadron Collider, where the Higgs boson was taken, you can take particles and make them collide. You study the aftermath, you may form something new in the process. Not interesting enough. You want to jack things up a bit, so you take bunches of particles and collide them together and watch the aftermath. So that's what the Large Hadron Collider actually does. But clearly I am talking about over the top things here, right? Bunches of particles, not exciting enough for me. Right? So let's scale things up. We don't call them astronomical scales for no reason. So how about we take 10 raised to 57 particles and collide them together. Again, hard to do that in the lab. That's more particles than exist on Earth or in the entire solar system. But the universe does it for us for free. Okay, so this animation here shows two neutron stars that are spiraling around each other, and you see these bands which are representing gravitational waves. And as these two collide, what you get is a massive explosion. And then there is a debris cloud created around it, large jets that were launched from the explosion, they go out and you get what are called short gamma ray bursts. Okay, and this is a scientifically informed illustration. So this is based on observations we actually did back in 2017. We as in astronomers from all around the world. The paper that was published about this had 3,500 authors. Okay, so very, very large, the largest collaboration in astronomy. And in this process, what happens is we end up doing something called R-process nucleosynthesis, rapid process nucleosynthesis, where now you have this ultra neutron rich environments and you collide those together, you form a black hole, but then the debris which was spread around, that is finally you spread some gold around. Okay, and how much gold, by the way? You have somewhere between order of 100 times Earth in gold. Okay, let's get that clear. It's not 100 times the gold that exists on Earth. If all of Earth was made of gold and you had 100 such Earths, that's the amount of gold that you create there, but it's not only gold. That is where you fill up the remaining periodic table, right? So that's where you are getting all of the lanthanides, actinides, and all of these heavier elements that you have. So we are all made of stardust, from stars which we killed not once, not twice, but thrice. So the universe really is a weird place. Now when you are talking about killing stars and deaths and so on, you clearly need detectives to go with that, right? So this is our team of cosmic detectives which probe this. This is our current iteration, but as I said, we have had more than 100 students working on this over the last 7-8 years who have all been working on Growth India and so on and other projects to actually piece together these mysteries. We already looked at Growth India. Here is the light polluted version of it again, since we have stage lights on, but that's just one of the tools that we use. The other tool that we use is a space telescope called AstroSat. AstroSat itself is some 8.5 years old by now, and it has 5 different, so this is India's own space telescope, and this has 5 different telescopes on top of it, but one of them, this cadmium zinc telluride imager, our team here is part of the core team that built it, okay, and we continue to operate that, we use it. So if I were to summarize the work done by my group, we build large toys to watch things go boom, right? That's the summary. And this is a particularly impressive toy for doing that. So we see these explosions around the universe. I used a phrase called short gamma ray bursts. Here is a bunch of gamma ray bursts that we have detected. We made this animation about a year ago when we detected 500 gamma ray bursts over a period of 6.5 years. So each of these dots, as you see them, and there are dates at the bottom, these are explosions that we have been sighting throughout the universe. I have the Milky Way superposed here, but these are not happening in our galaxy. The typical explosion that we are talking about is at least 1 billion light years away. So when I say that I saw it in June 2019, I mean that the light reached us in June 2019. The explosion happened 1 billion years ago, 2 billion years, 3 billion years, okay. So these are extremely ancient explosions. AstroSat has not stopped after 500. We have continued and crossed 600 last year. And then with these now, with a great instrument up there, unfortunately we don't always see everything. So I mentioned this event from 2017, and AstroSat is out there. It's one of the best instruments in the world to detect these explosions, but we did not see this event in 2017. And we calculated, and we were actually sensitive enough to see this. So why did we miss it? Okay, and here's what happened. So Fermi, a satellite, NASA satellite called Fermi, detected the burst, and it said, so this here, imagine it as a globe, but plotting the sky instead of the Earth, okay. And this brown circle here is what Fermi said the event must have happened, okay. And that's all they knew. So it's a large part of the sky. They could not pinpoint it. The exact position was found about 10 to 12 hours later when an image was seen. This is a galaxy and this is a source which did not exist earlier but was later found. So this was the explosion which was captured, and that happened to be located somewhere here, okay. And then this circle here, this is the part of the sky that we could not observe from AstroSat at that instant. So here's what is happening. You have Earth and you have a satellite that's going around Earth. So at any point of time, because we are in what is called a low Earth orbit, about 650 kilometers above the surface of Earth, about 30% of the sky is hidden behind the Earth, which means that you are not going to be able to detect anything that is happening there, okay. So with AstroSat, this was missed, unfortunately. And then we said we don't want to fall for this problem ever again. So if you want to go forward and if you want to solve this problem, if you are searching for these, and also note that I am standing here in 2024 and talking about an event in 2017. That's because after that we have continued searching, our group and other groups around the world, and we have not found any such event after that. They are extremely faint. We got lucky with the 2017 event that it was nearby. So we now need to be ready. We need to be better. So what do you want? You want an order of magnitude higher sensitivity. You want a wide spectral band, you want to detect this no matter what happens, and then you also want continuous all sky coverage, right? You don't want the standard desi answer, 5 minute chai peene gaya tha, tab tak event ho gaya, right? You want your satellites to be always on, you want them to be in the right place at the right time. The only way to do that is to be at all the places all the time, okay? So that's what we want to do, and for this we have proposed this extremely ambitious mission called Daksha. Okay, so Daksha is a IIT Bombay led national collaboration. We work closely with PRL, TIFR, IUCAA, RRI and various ISRO centers. And what we want to do is build not one but 2 satellites. These 2 satellites will use a variety of technologies and detectors to cover the entire sky. This is not what you would think a telescope should look like, okay? It's a dome. And the reason why it's a dome is that it is essentially a sphere covered with detectors so that we can observe anything that happens anywhere in the sky. So think of this more like the CCTVs that you have, which you also see only as a black hemisphere, right? So this is our sky CCTV, if you want to call it, but working in X-rays and gamma rays. And then we want two of them on opposite sides of the earth so that we can continuously cover the entire sky. All of these detectors are things which have been used in various Indian missions before. So you have heard about AstroSat right now, you have heard about Chandrayaan, I am sure. These are also used, by the way, in Aditya L1. These have been used in various missions as well. So this is all for making sure that we are extremely sensitive and extremely alert at all times for high energy transients. So if you want to be alert and if you are observing who's doing that job right now, so the current world leaders are these two satellites called Fermi and Swift. Fermi is a joint NASA-ESA mission, Swift is a NASA mission. We heard about the brown circle, which was Fermi localization. But the sensitivity of Daksha, or technically what we call the volumetric reach of Daksha, is going to be 5 times better than Fermi and 13 times better than Swift. Okay, and this is not a random tall claim that I am making here. The way you establish things in science is through refereed publications. This is all published in an international peer-reviewed journal. In fact, this very claim is co-authored by the person who's the boss of this mission. Okay, and all of this, we want to build these satellites right here at IIT Bombay, and we are right now at an advanced stage, but we don't have full approval just yet, right? Now to get to building a space telescope, there's a lot of activity that you have to do behind the scenes. You can't just wake up one fine day and chalo aaj ek telescope bana. Doesn't work, right? So all of these activities, all of these things that you have to do behind the scenes, you actually have seen all of them happening at IIT Bombay. So here is, for example, the main assembly of Daksha, what we call the dome, and this has to be something that is lightweight but extremely strong, because when you are launching a rocket, you actually shake things up very badly. The usual solution to that is you make things very heavy, extremely robust, but then the launch cost goes up. And not just that, for an assembly that is so complicated, you still want it to be easy to assemble and you want to have access. Once I have mounted all of this onto the central dome, right? Let's take our typical lab submissions as an example. There is always a loose connection which comes up when the prof is inspecting your demo or setup or have your viva, right? Now imagine something like that is happening. I want to access this panel, I need to have it accessible. Haath kahan se dalunga, repair kahan se karunga, and all of these things have to go into the design. We also have to worry about thermal management. We want to make sure that these detectors don't get too hot or too cold. So for example, when we first did our designs, here is what we got. Okay, so these are things which are a bit hard to read, but what is plotted here is as a function of time, as the satellite is orbiting around the earth, how hot or cold with different phases get. So the peak temperature here is 78 degrees Celsius, and the lowest temperature that you get across at that time across the satellite, you have a change in temperature of about 35 degrees. And each individual surface, the temperature was oscillating by about 20 degrees. You clearly can't work this way. Okay, the reason why this is happening is something like this. You have the satellite which is in orbit around Earth. So from this side I have the sunlight which is shining on this side. From here the Earth itself is radiating, so that will shine on the back side of the satellite. And from here there is no heat input. Plus there is power that is being consumed on the satellite itself. So when you put together all of this, you get an extremely complicated thermal problem, and then you want to go from this bad to something that is good. So we did all of this optimization, and then we finally started getting operating temperatures around 0 degrees, which is what we want to achieve. All of this is possible only at a place like this because we have engineering departments that are absolute experts in their areas. So all of this is done by collaboration between faculty from Department of Physics, Electrical Engineering, Mechanical Engineering and Aerospace Engineering, right? So that is how we can actually start designing and assembling something like this. And when we start putting all of that together, what we get is a satellite that's going to completely dominate this for the decade to come, right? Then there is development of detectors. So here's the setup. This again was done in elec and in physics. And with this, we actually can get, first we start by just looking at noise, that's how you always do things. But from there, we have now gotten full detector assemblies. These have all been tested, all been demonstrated to ISRO. And all of this work is something which is done with heavy student involvement, right? But just student involvement is not enough, right? You want to actually take it from basic project stages to something that can actually become scientifically acceptable standard around the world. So I'm just going to take a small detour here and show you switching from growth and Daksha to some projects that students actually have accomplished here. Okay, so for example, this is something which, so study of asteroids that was done. Two students, undergrads from Department of Meta and MEC actually discovered this asteroid, which was the closest asteroid to fly past Earth without actually hitting it. And then this is an image movie of another asteroid that we have tracked with the Growth India telescope. We have many students who are studying this. This I have not updated actually. We have tracked more than 200 asteroids plus comets so far. Okay. These students, a PhD students and the two undergrads actually won the best thesis, special mention in a new discovery award for discovering that particular asteroid. And then this year, this student who is now graduated and gone to Harvard for his postdoc, he actually won the best PhD thesis in astrophysics in the entire country, right? So this is all work that is being done here. I talked about AstroSat discovering gamma ray bursts, and I showed you a movie of these bursts happening across the universe, across the entire sky. Here is what they look like. So you are measuring some brightness as a function of time, it's jumping around, varying, and suddenly you get a big spike. Now all of this also was done by undergrad, and these are all students. You can see that the background doesn't matter. I have students from all departments. We have had students from a lot more than just this. We have had students from chemistry, CS, energy science, IDC. Okay, so it's all students coming together and putting their heads together to decide to do something amazing, and then this is what is possible. And on top of all of that, it's very important to remember that while we talk about academics who are doing this, it is not just academics working in vacuum, right? Physicists like to model everything as a spherical object in vacuum. You may have heard of jokes like spherical cows in vacuum. But we don't work in vacuum. We work in this institute, and the institute as a whole enables all of this. So I want to take this opportunity to thank everyone. This is about the administrative staff, the accounts people, the purchase people, outreach, alumni, everyone who gets together. And without the back end being robust, we can't actually have anything like this. For example, right now, without the nice teams from PRO and AV and CD and so on being present, we would not have been able to suddenly switch lights off in between to see what is happening. So science always is a collective endeavor. Everyone has to get together, everyone has to work towards a common goal, and it is only then that we can actually unlock the secrets of the universe. So I'll stop with that. Thank you.
Thank you, Professor Bhalerao. Do we have any questions?
So, I have curious questions. Now in CERN, the collider has gone to an energy about 13 TeV. So it's a basic question. Will this can we produce gold in the collider or not?
Okay, so rather than CERN in particular, let me take a more generic thing that can we actually make gold now in the 21st century? Are we able to manufacture gold? The answer is yes. Okay, but it is far more expensive than mining it out of Earth. Okay, so you'll produce a few atoms. You can start with elements which are nearby. You can bombard them with subatomic particles, and you can trigger reactions of either fusion or fission, which will occasionally give you gold atoms as a consequence. But it is far more expensive than what the current market values will offer. So strictly speaking, the answer to your question is yes. Would I want to do it? No.
So as you have mentioned that iron is having the maximum nuclear stability, but the origin of all elements is basically the hydrogen. So any other, for example, element which has atomic weight greater than that of iron should not then form. So how do they form actually?
So that is where the explosions come in, right? So if I want to do something in steady state, if I want to operate in equilibrium, then the minimum energy is what matters. But if I am not in a state of equilibrium at all, so when we have these explosions like supernovae, what happens at that point is you have an extreme injection of energy that is happening there. So in this explosion, you have now temperatures which were at millions of kelvins go to hundreds of millions of Kelvin. You have particles which are moving about relativistically, and they will go and collide into other atoms. And just like his accelerator question, they will form higher atoms, right? Now, the question as to whether we will find those atoms depends on the stability, okay? So, you have scenarios like this where, for example, technetium technically could be formed here, but there is no known stable isotope of technetium. So, it is going to decay away immediately. Whether we have observational proof for this. So, for example, in the remnants of supernovae, we regularly see lines of calcium, right? And calcium, you can't march up from here to create calcium. The way our theory tells us calcium should be created is by radioactive decay of titanium, okay? And with X-ray telescopes, so with an X-ray telescope called NuSTAR, we have actually seen radioactive decay lines of titanium in outer space, right? So the kind of experiment that you would do in a lab here, where you are sitting with a radioactive element, we have seen lines at 79 and 68 keV, which arise from radioactive decay of titanium. So we know this was formed in the supernova, right? So we have actually gotten observational evidence for a lot of this. Nickel, for example, is also quite close in stability to iron, but it does decay back into iron, and that is how we know supernovae are powered. So they rise up quickly, but they fade much slower, and that is because while the explosion injected the first energy, there is continuous radioactive decay of nickel that is happening, that continues for forever actually, but gives significant energy for weeks, which keeps the supernovae going. There is a question in the front here.
Good evening, Professor Bhalenam. Indeed, valuable insights for last 45 minutes. This is just out of curiosity. I would like to know what is the difference between astrophysics and cosmology?
Astrophysics, very broadly, I would say cosmology is a part of it. So, study of any objects in space, any natural objects in space, let's say broadly, would be astrophysics. So, that would include stars, planets, galaxies, and the universe as a whole. Cosmology in particular refers to the study of the largest scales of the universe. So, Big Bang, formation of early galaxies, what we call structure formation, and so on. There isn't a rigid boundary between them, saying that up to a scale of 10 billion light years is cosmology, smaller than that is astrophysics, or something like that is not the case, right? So, formation of galaxies is a part of cosmology, but is also a part of astrophysics.
I have a couple of more questions. I just googled the mass of Earth is some number into 10 raised to 24. So if we multiply by 100, that is the quantity of gold that you had mentioned.
Yes.
Yeah. That is one thing. Second is, do life exist outside our solar system?
I didn't. So the first one was a comment or a question? What was the question?
I just confirmed that Google
Yes.
said yeah.
Yes.
So that was.
Okay. And the second question is?
Do we have life existing outside the solar system?
We don't know. Right? That's the answer. Is there life outside the solar system? We don't have any evidence whatsoever of life. But that becomes a very tricky question to answer. Okay, and the reason why it is difficult is how do you define life? Okay, and the answer invariably comes down to we are implicitly asking about life as we know it. Okay, for example, if you try to go with a textbook definition of life, you would say that an organism that self-replicates under appropriate conditions, right? So my laser pointer is life because given appropriate conditions, also known as a battery here, photons are replicating themselves and coming out as a coherent beam. But you wouldn't say that this laser is life and I can kill it and I can start it again. Right? So what is life? We now have self-replicating robots. Is that life? No, then they have to be sentient. How do you measure sentience? You can't. Okay. So whenever we are talking about life in the universe, we have, and strict definition apart, intuitively we have a good grasp. This is alive, this is not. Right? So let's go with intuition and then say, okay, with this intuition, life as we know it, life of our sort, biology in some form, we believe that it has to be carbon based because you want to be able to form complex molecules. We also don't know any other solvent as good as water. So we also think that water will play an important role. So we search for organic molecules and we have found organic molecules, we have found alcohols, we have found all sorts of organic molecules in space. We have found habitable planets, we have found Earth-like planets at an appropriate distance from a star where water will exist in a liquid form. You want it to be in solid, liquid, gaseous, all forms so that reactions can happen. So we have found all of those, we have found precursors, but we have not found any sign of any civilization yet. By the way, also when we talk of life, we talk of civilization, but that's a huge leap from just having life. You can have microbes which never evolve into a civilization, and that is still life, and that is life as we know it as well. We have not found signs for that either. All of that said, I would be very disappointed if we are the only ones in the universe. So there's hope, we'll look for it, we don't have it just yet.
Thank you so much.
There's a question right next to you.
Do they take the help of software to see if the light is going down, because with the naked eye it is not possible to capture so many dots, and
Yes.
what are the algorithms goes, and why the particular person is given the credit when the program is actually finding the
Lots of interesting questions. So let's go after them one by one. How did I know that there was going to be an explosion happening here? I didn't. Okay. So this is like going for, say, bird watching. You don't know where the bird will be, you know there will be birds, so you keep looking. So these are what we call astrophysical surveys, and our telescopes go through pre-programmed or manually sometimes. We go and point at different locations and we keep looking at these galaxies every night, and then we compare the images, image taken today with image taken yesterday, and we see if something has changed. Okay, and that is how these discoveries are done. So when I introduced the growth network, for example, I said that this ZTF is our discovery engine. This telescope has been specially designed to be a very wide field telescope. Okay, so a typical telescope like Growth India is also a wide field telescope. So if you want to look at how much of the sky Growth India can see, let me see if I have a slide about it here. Sorry, I don't. But we see something that is about 0.7 degrees across. Okay, so full moon for reference is about 0.5 degrees across. Now, typical telescope will see 0.1 by 0.1 degrees. Okay, so 0.01 square degrees. ZTF can see 40 square degrees of the sky at a time. Okay, the sky itself is 41,250 something square degrees. So in 1,000 images, ZTF can cover the sky. A typical telescope will take something like 4 lakh images to do this. Okay, so we use these survey telescopes to find it. And even when you take these images, when you take these before and after images, you can't simply say A minus B equal to C. They are not numbers, they are actual data. So let's look at this movie of the Growth India telescope again. You will notice that this star is changing in brightness, but look at all the other stars, they also seem to be changing. Right? And that has to do with changing atmospheric conditions night after night when we are observing. So we have to work very hard to make sure that while subtracting these images, they are matched properly, they are scaled properly. The telescope, you see suddenly a white line appearing here because the telescope pointing was a bit offset. So we now have to align those images properly, and all of this work has to be done, and that is what these things are, right? And the software didn't pop into our lap, we had to write this. The software did it, but we made the software. So whoever made the software gets the credit for that. And we are very, very particular about this. So if you happen to sit at the telescope when something happened, does not give you credit for that. What was the work that you actually did? And then even after all of this automation, there is a database entry that comes here. That is where the creative part of science starts, because now you have to interpret the data. Okay, so with the advantage of an automated telescope, first is that we have had the core team which has worked very, very hard to make the telescope automated. Right? We have more than a dozen telescopes in the country, only one is automated. So it's not easy to do that. And once that was done, now when that data comes in, our software now gives us the brightness of this object. So essentially I'll wake up in the morning and I'll have a table which says that at 12 o'clock I observed this object, here is its brightness. At 12:05 I observed this object, here its brightness. So I have a table which gives me brightness versus time. But then I have to go ahead and interpret it. That is the creative part of science. And that is where whoever is leading that analysis is the one who gets the credit. No point for getting lucky.
With the modern technology like artificial intelligence, are we feeding this database to the AIs also so that they interpret it in a maybe more accurate way, or we don't miss any?
Yes and no. Okay. First is, so this is a pet peeve of mine. Artificial intelligence is not magic. Okay, and it's going up right now, but it's not that new either actually. ML has been around for a while, and what used to be called ML a long time ago is now called regression, and then our tools are getting faster, and in astronomy we adopt technologies very fast. So virtual observatory databases, the largest big data sets in the world first started with astronomy and astrophysics. Right, so we do use all of these things wherever they are applicable, but it's not black magic. I can't toss data and say isme kya hai, right? And you could compare it with natural intelligence. There are data sets which have existed there for a decade, and then someone bright comes in and has a new insight which no one else has. Right? So I'll refer here to Hitchhiker's Guide, saying that artificial intelligence will tell you 42, but you don't know what the question is. Right? So to get the right answer, you first have to start by asking the right questions, and that is where again I am saying the creative part of science comes in. You need to have a broad understanding of the field. You need to ask figure out what is a question worth asking and answering. And once that is done, in these parts somewhere here, we actually have, so this perform subtraction, and then we have this, and then there is human scanning. So this step, parts of this are done by a machine learning code. Okay, but it is still a machine learning code that has been trained by humans. So it is still only as good as we can make it. You'll say, oh, what about deep learning? What about this? Yeah, those things are being tried. And there are things which are coming up, but also we have to remember in these days it is very easy to think of artificial intelligence as solving all problems, but it has actually become so large already, computationally so expensive, that it is one of the major contributors, for example, to greenhouse emissions from astronomy. So you want to do algorithmically what you already can before you go that way. So there are specific parts where ML and AI are useful for our work and we use them there, but everywhere else natural intelligence works better.
Thank you very much.
We have one more question.
Thank you for the great talk. You compared Daksha with the best missions out there that are currently by NASA and other space agencies. How does Daksha compare with Astrosat, which looked like the best that we have done so far?
So comparing Daksha with Astrosat in this context, Daksha is far, far better, easily 10x or more. But it is not a fair comparison for Astrosat, because in some sense you are comparing, to give an analogy, you are comparing a batsman with an all-rounder. Okay, Astrosat is an all-rounder. There are many good things that Astrosat can do which Daksha cannot, but Daksha is not aspiring to do those either. Astrosat is for simultaneous multi-wavelength observations. Daksha will do broadband observations, but only in X-ray gamma ray bands, for example. So there are some things which Astrosat will do much better, which Daksha is not even trying to do. But in terms of finding transients, which is the core job for Daksha, it will far outperform Astrosat. The lifetime is about 2x better, the field of view, the localization, the sensitivity, everything is order of magnitude better.
Thank you, sir, for the great talk. My question is why the telescope is situated in the Ladakh? Because the place is very remote, you hardly get electricity, you hardly get access to the internet.
Yes.
So why is
those are all very, very valid problems that we actually have to worry about. And I'll give you the answer very simply. I could put the telescope in Mumbai, and then I would see
Mumbai,
this.
so let's say a more cleaner place, more cleaner skies.
Correct. Yeah. So how do you find such a, no, keep the mic, because I need your help in finding that place. How do you find such a place? Where do you go?
More altitude.
Correct. So where do you have tall mountains?
Yeah, obviously in Himalayas.
But so, okay, now I'll give you my side of the answer. We as in Indian astronomical community did site surveys everywhere. There are telescopes in Kavlur, there are telescopes on Pune-Nashik highway. On one side we see GMRT, which is on the plains because it's a radio telescope. It doesn't need to be high altitude. But on the other side there is a mountain called Giravli, which has an optical telescope. telescope in Mount Abu, there is a telescope being set up in Bengal only about now. There are telescopes near Nainital in Devasthal, and there are telescopes in Ladakh.
Yeah.
So people went and spent years at these sites setting up equipment, measuring the quality of the sky. We actually can quantify it. We can measure how bright the sky is. We can measure the turbulence which affects our observing conditions. We can measure number of cloudy nights. Right? So Kavlur, or why is the telescope in Bengal coming up only now? It's coming up also mainly as a teaching and training telescope, because more than half the year it is clouded out. So it doesn't help. So doing all of this, you need to go to a site that is remote, that is high altitude, because lower altitudes you have a lot of water vapor, and water vapor absorbs infrared, and we also want to use these telescopes to study the stars in infrared, so you don't want that. Lot of dust near any human settlement, lot of light pollution, you don't want that. So you want it to be remote, right? And then, come on, we are all IITians, we are lazy. You don't want to go to a remote place and work. That's why we automate it, right? Be efficient. Be lazy, but call it efficient.
Thank you.
So we sometimes call this deferred laziness, right? We worked hard for the last few years so that we can be lazy for the next few years. The telescope is now doing its own thing, and now we can sit back and enjoy the science.
Thank you so much for the thrilling talk. So my question is a bit, it was something back in my mind for a long time, because I myself work in particle physics. So I want to understand what is the basic difference when we talk about particle physics and astrophysics. Say we both are trying to understand, ultimate question is like the beginning of this everything. So what is the fundamental difference between a particle physics approach and astrophysics approach?
This is my snarky answer, which is, for such small number of particles, they are much smaller than error. They are smaller than the error bars on our error bars. Okay, okay. But the main difference here is that astrophysics, as I said, is about things in space. Right? We are trying to understand things like equation of state, yes, and we got great constraints from 1708-17. Understanding the nuclear equation of state at supranuclear densities, with 3 times nuclear densities, for example, can only be done through astrophysics, because in accelerators we can't reach pressures or densities that high. Mass radius relationships of neutron stars, all of these things give us some particle physics inputs, but we also study why there is dust in the galaxy. We also study whether we are going to find life in the universe. So astrophysics is a very broad field. Similarly, in nuclear and particle physics, there are aspects that you are studying which are not relevant to astrophysics. So there are areas around, for example, equations of state, around nucleosynthesis, which are overlaps and intersections between the field, but the field by itself is completely different, right? Now, interdisciplinary nature happens in all areas. If you are trying to, for example, in the last talk, we talked about medication, which then connected to mental health, but then if you go on this side, you want to connect to manufacturing medicines, which now goes into engineering, right? So anything that you do, any modern discoveries, modern technology, all is interdisciplinary. So if you want to reach these kind of densities, you can't do that in the lab. We can reach those, but at the same time, you couldn't find a signature of a Higgs boson in a binary neutron star collision, because the collision is so chaotic. You are giving out something like 10 raised to 50 ergs of energy in a few seconds. Okay, astronomers use weird units. Okay, so convert to joules by yourself. But you are giving off these intense amount of energies, and in that chaos, you don't have your grid of trackers sitting around to tell you every particle that went out, because while I can say I have 10 raised to 57 particles, it also means I can't track all of them. So there are areas where inputs from here come here, inputs from here go here. One of my past students who went on to do her PhD in equation of state and related field, her work for astrophysics was calculating the electronic levels of all the lanthanides and all these elements which are formed in neutron star mergers, because they will help us interpret the variation of brightness with time. And turns out particle physicists didn't care, because lab mein who cares ki lanthanum excite kaisa hota hai, right? So there are all these interdisciplinary areas that we need to work on, but the broad focus is not the same. Does that answer your question?
Yes, exactly. And also I have a comment regarding the question, the first question that was asked, like at CERN currently we've been able to reach center of mass energy, say 13 TeV. Now is there a possibility for formations of gold directly? So the questions is, sir said, of course it's possible, but the thing is, even if you create a kind of state, say, with the collisions of two matter distributions, say nuclei, so it only lasts for a very small time. So essentially, in the final state, what you get is elementary particles, light particles, light mass particles. For the formations of gold, you itself need a system to last for a longer time, actually.
No, that's why I said not in the context of LHC, right?
Yes,
LHC, you are
exactly.
starting with elementary particles, you get elementary particles.
Right,
but
exactly.
for example, if you go to TIF or Spelletron, you do actually synthesize other elements by bombarding normal elements with alpha.
Yes,
so
yes.
The answer that I gave, that is why I avoided LHC in particular.
Yeah,
You can synthesize elements, it's just a very impractical way of doing it.
Okay, thank you so much.
Thank you. I think we are short of time, so if you have any further questions, you can connect with Professor Bhalerao.
Thank you.
I'll begin with a vote of thanks. Thank you, Professor Maji and Professor Varun Bhalerao, who graciously agreed to deliver the lectures at such a short notice. I would now request Professor Upendra Bhandarkar, Associate Dean, Research and Development, to present a memento to Professor Maji and Professor Varun Bhalerao as a small token of gratitude. Professor Varun Bhalerao. Thank you, Professor Bhandarkar. Many thanks to all our guests present today. I also wish to thank my colleagues from various sections of the Society of the Institute for coordinating and organizing today's lecture. Thank you once again. Please join us for the refreshments.